Acquired immunity. Immunity acquired by infection or vaccination or by the transfer of antibody or lymphocytes from an immune donor. Passive immunity. Acquired immunity is in contrast to innate immunity or natural immunity. Specific immunity, also known as adaptive immunity, is specialized immunity for particular pathogens. Helper T cells, cytotoxic T cells, and B cells are involved in specific immunity. The non-specific cells like macrophages tell the T and B cells that an intruder is present. Endurance, development, and self-recognition also important. Active and passive immunity. Active immunity, the protective immunity in which the individual's own immune system is stimulated to produce antibodies and lymphocytes. Passive immunity, the immunity in which a person receives antibodies or lymphocytes that have been produced by another individual's immune system. Cell mediated immunity. Cell mediated immunity is an immune response that does not involve antibodies. Rather, cell mediated immunity is the activation of phagocytes, antigen specific cytotoxic T lymphocytes, and the release of various cytokines in response to an antigen. एंटीमाइक्रोबियल सेल मीडिएटेड इम्यूनिटी जीवाणुओं द्वारा उत्पन्न संक्रामक रोगों के विरुद्ध प्रतिजीवाणु सेल मीडिएटेड इम्यूनिटी का उपयोग में आते हैं. एग्जाम्पल माइक्रोबैक्टीरियम ट्यूबरक्लोसिस, बैसिलस जनित रोग ट्यूबरक्लोसिस से रक्षा सेल मीडिएटेड इम्यूनिटी द्वारा होती हैं. नॉन इम्यून होस्ट में संचारित होने के उपरांत इस रोग का जीवाणु मैक्रोफेज द्वारा अंतःग्रहित कर लिया जाता है तथा यहाँ यह गुणन कर वृद्धि करता है. अतः इस रोगाणु को नष्ट करने के लिए एंटीबॉडीज प्रभावी नहीं होती. एंटीवायरल सेल मीडिएटेड इम्यूनिटी, वायरल एक्टिवेशन ऑफ इम्यूनिटी, local infections at surface such as the mucosa can initiate local cell mediated and humoral immune responses but not necessarily systematic immunity. Cytotoxic T lymphocytes, natural killer cells, and antiviral macrophages can recognize and kill virus infected cells. Anti-tissue cell mediated immunity. Once they have completed their development in the thymus, T cells enter the blastoderm bloodstream and are carried by the circulation. On reaching a peripheral lymphoid organ, they leave the blood again to migrate through the lymphoid tissue, returning to the bloodstream to recirculate between blood and peripheral lymphoid tissue until they encounter their specific antigen. Mature recirculating T cells that have not yet encountered their antigens are known as native T cells. To participate in an adaptive immune response, a naive T cells must first encounter antigen and then be induced to proliferate and differentiate into cells capable of contributing to the removal of the antigen. We will term such cell armed effector T cells because that act very rapidly. When they encounter their specific antigen on other cells, the cells on which armed effector T cells act will be reformed to as their target cells. Antibody mediated immunity और humoral immunity and cell mediated immunity के अंदर differences. Definition. The immunity mediated by macromolecules found in the extracellular body fluids is called humoral immunity, and the immunity that identifies and destroys infected cells in the body is called cell-mediated immunity. Mediator humoral immunity के अंदर the main cell involved in the humoral immunity are B cells, and in cell-mediated immunity the main cell involved in cell-mediated immunity are T cells. Components B cells, T cells and macrophages in humoral antibody. Helper T cells, cytotoxic T cells, natural killer cells and macrophages are in cell-mediated immunity. Pathogen recognition recognize antigens or pathogens that are circulating in the lymph or blood, and it responds to any cell that display aberrant MHC markers, including cells invaded by pathogens, tumor cells or transplanted cells. Accessory surface receptor immunoglobulin A, immunoglobulin B, Fc receptor CD14 and CD21, and in cell mediated immunity CD3 molecular complex dimer of epsilon chain, CD4, CD8, CD2 and CD28 integrins. Humoral antibodies में antibodies formation in a humoral response, and in cell mediated antibodies are not formed in cell mediated immune response. The onset is rapid and the onset is delayed. The end result of the activation is the differentiation of plasma B cells and secreting antibodies in humoral antibodies, and in cell mediated antibodies the end result of the activation is the secretion of a cytokines. Role in organ transplantation and grafting in humoral antibody. It may be involved in early graft rejection due to performed antibodies, and rather than in cell-mediated immunity, it participates in rejection of organ transplantation. Assessment methods in humoral antibodies from plasma level of antibodies, and in cell-mediated immunities, it's a skin test for the development or delayed type of hypersensitivity. Thank you.